Yeah, so I'm a biologist by training now, so no equations this time. And as you can see, I'm trying to give more a general overview about migration of epithelial cells, so of cells that depend on focal adhesions and also on filopodia for migration, especially because for the next days we have for a couple of those aspects I'm trying to deal with more specific talks later on. So, migration of epithelial cells. Before I start, actually, I'd like to mention that basically every cell, especially if you isolate them and put them in cell culture, is almost able to migrate on the surface. So here, for example, embryonic fibroblasts, and you will see in a second over time some cells of them are some kind of polarized, some of them are just very dynamic, and if you analyze those cells, you see them migrating on the surface, but and you can also track this migration over time in an XY coordinate system, and what you see then is that those cells certainly migrate in all directions. The persistence length of those cells is not really very high, and it's then just a question of time. If you would like to have a cell migrating from a position here, for example, at 0 to some other position, until this actually takes place. But, however, with such a migration, very specific structures, as for example here during zebrafish embryogenesis, wouldn't really be possible. You will see here this migration is a very well directed migration of cells forming the neural tube. Very fast, very specific, and perfectly well directed. And about this directed migration I will talk within the next approximately 45 minutes. You've seen this here already before, because this directed cell migration very much depends on mainly two structures that are the focal adhesions and also on the actin cytoskeleton that is bound to the focal adhesions. And then in this stepwise process, new extensions are getting formed, forming the lamellipodia we have already heard in the former talks. These lamellipodia then are able to newly attach to the surface, form new adhesions, and then by time the cell contracts by generating forces. The whole cell body becomes transferred in direction of cell migration, and then finally at a step where already very often new lamellipodia getting formed, so it's a continuous process, old adhesions are getting disassembled. This is here now on the right a scheme for epithelial cells. This is how those structures really look like. You have the cells well polarized. In this case it's a keratinocyte, so it's a skin cell of mammals, and you have this well polarization. It's basically a half circle. Here you have the lamellipodium. This cell always migrates in direction of the half circle, as we have already seen in those movies. And here on top you see the adhesion structures, and here at the bottom you see the actin cytoskeleton. At the end of the talk you will see all this more dynamically, and hopefully will understand how the whole thing is regulated in principle. So when I talk about epithelial cells, in this case keratinocytes, the first question that arises is how a cell that is typically completely sessile, forming, for example, the dermis, so the skin, suddenly can become motile. So this is something really that is, when you check your skin, not very obvious, because those cells in the different layers are well attached to the basement membranes by cell matrix interactions, but also to the surrounding other cells by cell-cell interactions, and even the composition and the structure of the cell-cell interactions then varies from layer to layer. But this very stable structure becomes immediately very motile upon a scratch, a wound, inducing the motility, the migration of those keratinocytes, epithelial cells, by specific chemical factors. And those chemical factors then make sure that the cells really induce a directed migration in direction of higher concentrations to form then beneath the fibrin clot new skin, and so that the wound is getting closed. This directed migration, we have seen those movies already, so biologists like those movies as well. So I'm not necessarily showing this movie. Those migrations you see here for macrophage, or it's a neutrophil actually, of dictyostelium, where you also have those directed migrations, always depend on a gradient of specific chemicals. Okay. Oops. So in this case, it's a chemical of LPS, in this case of CAMP, and the cells then are tracking the whole system. In the case of keratinocytes, epithelial cells, you also have one gradient, and this gradient is growth factor, endothelial growth factor, that becomes formed by the cells surrounding the scratch or the wound. So you have a gradient, and the EGF, which is then placed in the wound, becomes recognized by receptors. Those receptors then dimerize, and this here now is what's going on within the cell. It's not really important to understand it in detail, but the most important thing is that external signals become incorporated into the cell, and then leading to a couple of changes on the level of actin reorganization, cytoskeletal rearrangement, and focal adhesion assembly and disassembly. And this is what then comes out at the end. The keratinocyte that was originally sessile then migrates with a very long persistence length in one direction, and you see here again the retraction fibers at the end. Here up front, the lamellipodium with those tiny little extensions. You will see them later on even better, the so-called philopodia. And this system then migrates in direction of higher EGF concentrations.
But what are the filaments that are in the back? So they are parts.
So they also contain actin, and they are especially connected to remaining adhesion sites, so basically integrins, and most of those integrin molecules that are still attached to the surface even get ruptured out of the cell. So they remain, leaving a migration track behind. So then a lot of those retraction fibers also remains bound to the surface, or is getting brought back to the cell body. So schematically, such a migrating keratinocyte is shown here. Here a real trailing edge. Sometimes you just have, as shown before, those retraction fibers. And I would like to go now through the four most important areas in the cell leading to directed cell migration for epithelial cells. And this is, we have heard about it already before as well, the leading edge, then in addition the philopodia, then the focal adhesions that very often depend in formation on the filopodia, and then also I'd like to mention the lamellar. So there a lot of different regulatory aspects are going on. So let's start with the lamellipodium. So within the lamellipodium, as we have seen, the actin biopolymer constantly polymerizes at the plus end and depolymerizes at the minus end. So, and by this, it is able to push against the plasma membrane, pushing this membrane in direction of migration. However, since this actin filament has a width of approximately 7 nanometers, it is relatively stiff if you check a small rod. But if you have a very long filament, then this filament attaches or connects the basement membrane, and that simply gets bent and cannot really push the membrane forward. For this reason, the cell was pretty smart and is using this ARP2/3 complex, which binds to existing actin filaments and leads to the formation of a new actin filament. So you get a branch caused by this ARP2/3 complex. With this complex, the cell is kind of cheating a little bit because, as you have also already heard, the actin in solution is hardly polymerizing on its own. You need to have at least 3 actin proteins combined to a first complex in order to start with the real polymerization, which does not really take place very often on its own. And by having this ARP2/3 complex, you simply get 2 actin-related proteins directly bound to an actin, and then just adding of a 3rd actin protein is sufficient to form this new branch. You see this here once again, here in red, the ARP2/3 complex, and then continuous branching of already existing actin filaments. It's approximately an angle of 70 degrees, and also very important, then those actin filaments are getting kept by other proteins to make sure that their length is not exceeding too much, and by this you get the classical actin structure within the lamellipodium. To make sure that this actin structure takes place mainly or almost only at the lamellipodium, some other proteins are very important, and that are small GTPases. You can see them here in red as well, that bind specifically to the membrane upon activation, and those GTPases then induce more or less the activity of the ARP2/3 complex wherever the GTPase is bound to the membrane. And since this binding again depends very much on other receptors, the binding of the of the GTPases takes mainly place at the lamellipodium, and therefore in this direction also the polymerization takes place. How it then really looks, you can see here on this image from Svitkina and Parisi. So it's for me really a very fantastic electron micrograph, and you see here the actin filaments then continuously branching, and by this the actin network can push the membrane effectively in direction of migration. The forces that are getting produced by those actin filaments are relatively low. Here, for example, those experiments were made by having a bead in a laser trap, and from this bead then actin filaments are polymerizing against a wall, and as soon as they reach the wall, they push the bead away from the laser trap, and by this you can actually analyze and determine the forces, and they are in the range of approximately 1 piconewton per actin filament. So they are pretty low, but of course a lot of those actin filaments push simultaneously.
If you back up in the cartoon that you drew as well as in the micrograph,
Yeah.
the branching seems to be happening in the same plane. Is that very much dictated by the geometric confinement in the lamellipodia? If you do this in vitro, would you be able to get branching all over the place?
Yes, you would. So if you would do this in vitro, you would get the branching everywhere.
True that in the lamellipodia is actually 2D?
Mostly, yes. So it's I would say mostly 2D.
But it still has like 200 nanometers thickness.
That's true,
right?
yes.
So in principle, somebody calculated 8 layers of actin, but they seem to stay in layers, right?
Yeah, so they are in layers. So this is why it is, I would say, very close to 2D.
And do you know the rate at which these branches are formed?
Sorry?
Do you know the rate at which these branches are formed?
So I think the average branch length is around 50 nanometers. So pretty small. And then the next branch starting. So the way then it looks, you see here, if you have a keratinocyte just attached to a surface that induces just a very tiny little adhesion. So low-coated substrates where the cells can nicely push the basement membrane at the lamellipodium, the cell membrane, and but are not really able to induce the formation of adhesion sites. By this, you get continuous formation of lamellipodia that are then getting retracted within the cell body. So as you can see in this movie, this cell is able to push the membrane in all directions, but there was no polarization of the cell, and there was also no migration of the cell. So something else must be necessary for this cell type in order really to get migration. And this is indicated here by the filopodium and then later on also by the focal adhesion. So the filopodium is something that starts with the structure you've seen before in the lamellipodium. Here you have the R2-3 complex, the branching of the system, and then at specific locations at some points, around 20 to 30 actin filaments become bundled by specific cross-linkers. In this case, it's fascin, and also proteins bind to the polymerizing end, to the plus end of the actin filaments, inhibiting the capping of those filaments and making sure that those filaments are able to grow over a longer period of time. So those proteins are formins. And by this, then filopodia are getting formed, extending the leading edge. Here you have the cross-linkers and the formins sitting at the plus ends of those actin filaments. Those actin filaments, also very important, always have the same directionality, meaning all are growing at the plus end, at the tip of the filopodium, and the minus end is within the lamellipodium. The way it looks, you can see here, you have the epithelial cells migrating over the surface. You see here the filopodia extending the lamellipodium, and what you also see is that they stably adhere to the surface. So this is very important. Some of them are getting pulled back, and some of them are really adhering very stably to the surface. You can see this here once again. just follow here the white arrow. We have here the filopodia, and those filopodia get extended, they bind to the surface, and then caused by the ARP2-3 actin polymerization, the leading edge follows the filopodia tip, and at some specific point then the filopodia becomes reactivated and elongates one further step. You see this here, binding, and you get a repetitive elongation of the whole system. And in between, those filopodia are really attached stably to the surface. The way they do this is indicated here, because the proteins you see here, paxillin, taline, vinculin, tensin, are classical proteins you find in focal adhesions.
Sorry, since you show so many movies with retraction fibers, are there any function in the retraction fibers? It's just like, seems like slowing down the cell.
So to my knowledge, so if there's a specific function for the retraction fiber. So just keep in mind, what you see here is a filopodium that's at the front. So this is not a retraction fiber. So so the retraction fiber, to my knowledge, are not really having a very specific biological function, except that cells are able to follow each other. So because they are left behind, a couple of specific proteins then are also left behind in this migration track. And then if one cell reaches or sees this track, then the cell follows the first cell. But
Migration.
sorry?
For collective migration.
Exactly.
If you think of a filopodium, is the membrane still there?
The membrane is still there. Yeah. So the width of the filopodia is approximately 50 nanometers, and the membrane is surrounded. So there's, I will come to this in a second. So because what you have in the filopodium is the localization of all proteins you also have in the focal adhesion. So meaning that those filopodia you see here are bound to the substrate most likely by some kind of premature adhesion complex. So in this adhesion complex is not only found in very classical, well elongated filopodia with a length of approximately up to 5, sometimes even 10 micrometers, but also in microspikes, so that are most likely some kind of very, very tiny, small, rarely visible filopodia, mainly also caused by the bundling of actin filaments within the lamellipodium. So, and now you have here the filopodia, and what you can see now is that directly behind those filopodia in the lamellipodium, focal adhesions are getting formed. So those focal adhesions here are shown by a specific microscopy techniques. This reflection interference contrast microscopy. In principle, all you need to know about this technique is that the closer something is to the substrate, the darker it appears, and since the focal adhesion is closest to the surface, you get the elongated focal adhesions well visible. So, and you see here such a scheme of a focal adhesion where you have the extracellular matrix. This is basically what we have on the glass surface, and then the cells binds via integrin heterodimers to those extracellular matrix proteins, and they connect the extracellular matrix to the inner actin cytoskeleton by a bunch of up to 150, at least at some point, proteins within the cell. So important is that the localization of those adhesion structures really depend on the filopodia in many cases. Because the first adhesion I mentioned before, where you have all those proteins, simply is already existing in the filopodia and just become enlarged, forming a real focal adhesion as soon as the leading edge overgrowth the filopodial actin bundle. So indicating that the whole system here, the filopodia, is also bound via integrins to the surface. This is why then the whole system just becomes enlarged in size within the lamellipodia, and therefore, since integrin is a transmembrane protein, the whole filopodium must be surrounded by membrane.
So is it breaking down, or is it a continuous filopodia, or does it break down at some point and regenerate?
So the first formation of a filopodium is really induced, most likely, as I showed, by the bundling and then the formin formation. And now, as soon as the filopodium, yeah?
So the forming, well, at the beginning, the cap, I don't understand. So you have first the polymerization, and after the polymerization of several filaments in parallel, and after you have the adhesion, or you have the adhesion triggering the polymerization. I don't know I don't understand what triggers what, and if the forming is synchronizing the polymerization of the several filaments, I don't understand.
Yeah, so what you have is, so a couple of questions are still unclear to my knowledge. So what you have is, at first, the bundling of actin filaments within the lamellipodium. So formin binds to the plus end and induces the polymerization of all those actin filaments, and then they elongate, forming the filopodium, and at some point this filopodium binds to the surface. It is kind of sensing the surface for best adhesion. So what makes the filopodium stop polymerizing is an open question. So it is still not absolutely clear. It might be that it is driven by specific regulations. It might also be that it is driven just by a limited diffusion of G-actin all to the top or the tip of the filopodium. So both is actually possible and still unclear. And then this filopodium is adhered to the surface, and the lamellipodium, induced by ARP2/3 polymerization, follows the adhered filopodium. This is why then over time the leading edge overgrowth, the still existing actin bundle in the lamellipodium. And interestingly, for many cell types, especially for keratinocytes, then at some point well-adhered filopodia are not getting then completely overgrown and a new filopodium is formed, but instead the so-called tip complex at the tip of the filopodium becomes somewhat reactivated and polymerizes once again. So, and then looks again for new adhesion, and this is why in keratinocytes very often upon migration the adhesion sites follow one after the other.
There are more comments there, maybe Sasha.
No, I want only to a little bit restore the balance, because you are saying that focal adhesions are formed from filopodia exclusively.
No. And this is not what
as far as kind of, I think majority of people believe, there is a formation of focal adhesions from so-called nascent adhesion or focal complexes, which are not related to Philopodia. I cannot exclude that in some cases focal adhesions are formed by Philopodia, but it is rather special.
So I will come to this point, but I fully agree. So in this case, it is very important that the focal complexes are primarily formed within the phylopodium. But what's most important for the formation of a stable focal adhesion is a well-adhesion connection to the actin cytoskeleton. And then also later on, forces that need to be applied to those very young, premature adhesion sites. But I will come to this later on.
The Philopodia growing at the tip. So that there has to be then some transport of molecules to it. So where are those molecules coming from?
The molecules at the tip?
Yeah, but sorry, I mean, you got suddenly the appearance of a line like this, and everything is happening. The growth, the molecules are there at the tip. So there's going to be
it's a very good
transport of molecules.
It's a very good
to the tip of the Philip. Yeah, I don't know.
Yeah, so it's a very good question. So there are some motors migrating along those actin filaments, presumably bringing proteins to the tip. But most of it also seems to be just diffusion.
Fusion. But within the cortex,
Through the cortex.
Through the cortex.
Yeah.
So there's very, very fast diffusion in matter.
Yeah.
I mean, yeah, that's a puzzling question for many
Yes,
things.
it is very much a puzzling question. It
But
would
it's
be
very
very
fast,
interesting
but there's
to
also-
have a model for this.
But there's a massive but there's a really big mechanical deformation of the cell, isn't there?
Yeah, sure.
This is-
Of the membrane.
First,
Yeah,
yeah,
of the membrane.
yeah.
This is why you need also a bundle of actin filaments. They really need to be bundled to get a kind of stiff rod.
The molecules then have got to move up parallel to the Philopodia.
But some of them are transported too, right?
Some of them are transported, as I said. Yeah.
But they're not moving along the surface of the membrane.
No.
They're moving from the
So how do they
interior.
move? Yeah, exactly.
So I mean, there's two things. One thing, there's active transport also. That's what you were pointing to, right? So there's also motors which can transport things along the phylopodium. And the diffusion, I mean, there's models for like when the diffusion becomes limiting, and with the 200 nanometers meshwork, it's not really a problem for small molecules. It's a problem for myosin and things of that size. But although
Actin is not in there. So.
But Philopodia can get very long.
yeah,
What's
that's,
the,
Do you have any
you
sense
would
of
worry-
what controls the density of Philopodia along the
Yeah,
membrane?
the
Are there
transport
competing
becomes
processes that set the spatial scale, or what?
You mean how many Philopodia you actually
If you
have?
look at the left image, toward the front, you have a
Philopodia.
relatively regular
Yeah.
spacing.
So I have no idea. So we never checked. I don't know if anybody checked. Do you know if there's an analysis of the density of Philopodia?
Depends on cell type.
Yeah.
Density of Philopodia. Some cells do not form Philopodia at all.
Yeah, but I mean, within a given cell type, there seems like there are competing processes that set the spatial scale. But I mean, if you're in a growth cone in a endothelial cell or a neuron, you can have 50 or 100 Philopodium coming from a tiny area of membrane.
Of course, there are
So
huge
you can
differences.
get enormous densities of Philopodial protrusion. And they do compete with each other too. So if you have growth, there seems to be a competition for material to go into the Philopodium to extend them. So you typically get these growth cones where you have an angle of growth with the density highest at the two ends, and then depending on whatever signal they're somehow transducing from the environment they're seeing, you'll see a preferential movement of material from one of those groups to the other. But it's,
So which comes first? Is there a weakness on the membrane, localized as a point?
I don't think that is a weakness. So it's
Well,
really
no,
a-
because it's
it's actin pretty
an
much.
active regulation. It's really an
which
active
causes
regulation.
the growth which causes the growth in a line. or is it suddenly at a point on on a line perpendicular to the membrane, which is the direction of the filopodia? Suddenly there's a massive force.
There's also a number of proteins which specifically change the curvature of the membrane, which
so there is something happening on the membrane.
Probably needed in concert with pushing, so that you achieve this high curvature. Otherwise,
Yeah, but aren't you changing the balance between different signal transduction pathways as when you form a filopodium versus a lamellopodium, for example? I mean, you're turning on different pathways, right?
And there's also proteins which stabilize curved membranes, so like bar proteins. So once you start forming a bulge, you can have. But that's a very much active field, I think.
In other words, nobody knows for sure.
The force
I think there are
required to
papers by
protrude the-
Niergov, my colleague at Weitzman, that have tried to rationalize at least the thickness of the filopodia based on attractions of the filaments within the bundle and some kind of surface energies having to do with proteins or surface energies of the membrane against that the bundle is pushing against. NAI-GOV is the name.
Oh, sure.
So I think he has some theoretical papers that have tried to model and rationalize at least the thickness of one filopodia bundle. If not, he might also have stuff on the space.
But the force required to move the membrane is not very big. So if you're concentrating on the issue of membrane deformation, that's not the main force that the filopodium has to fight against.
And my question was, is it to do with what goes on in the cortex, or is it to do with what goes on
How is it
on the
not
membrane?
nucleated? That's the
I think-
question.
That's certainly a group. There
That's
certainly
the
are
question.
molecules that are membrane associated that nucleate filopodial formation.
But the short, I mean, I think that's probably a bigger discussion point, which we may want to keep for, because there's no clear answer.
No, it's good. It's good to discuss.
Yeah.
This is very good.
Yeah. Because there's force to deform the membrane, but there's also membrane curvature, which is very high, and that's a problem in the filopodium, which costs energy indeed.
But that also attracts proteins that then promote filopodial extension. Because they're curvature-associated proteins at the membrane. So there's a positive feedback
Okay.
for
You say there is something activating the R23, so something need to activate the forming.
The formin as well. So the formin becomes activated as well, also by partially small GTPases, and this then induces the activation of formin and then the formation of the filopodia.
Okay, so it's the different pathway we were speaking about.
So now let's come to the connection, as Sasha already mentioned, between the filopodia and the focal adhesions. I'd like just to show for this specific cell type the dependency of the focal adhesion formation or stable focal adhesion formation on filopodia, but later on like to mention that it is really just important for a stable focal adhesion to have the connection between an actin bundle or an actin cytoskeleton and including myosin and then the adhesion proteins. So what you have here is the localization of filopodia, and this area now will be analyzed over time, and you see that wherever a filopodia is getting formed, later on the lamellipodia, the focal adhesions become in place. So you have the filopodia formed, a focal adhesion. The filopodia formed, a focal adhesion, and so on. So this is, for this cell type, what makes sure that the stable focal adhesions immediately have clear contact to actin bundles. So this is what it's all about in principle. So while in other areas where you don't have filopodia, actin bundles, and just the R2-3 meshwork, there you get also adhesion contacts, but those adhesion contacts do not last very long because no force can be applied to those adhesion contacts, and they disassemble over time. And the strong signal you see here is actually again formed by a phylopodium. See this here once again. So indicating that there's really, for this cell type, a clear correlation between philopodia and focal adhesions. And of course, just analyzing one single cell and a few focal adhesions doesn't mean too much, but by analyzing hundreds of adhesion sites that were formed in a philopodia-dependent manner compared to adhesion speckles formed in a philopodia-independent manner, you can really see very well that the size of those adhesions formed here in light gray, the philopodia-dependent ones, is much higher than the adhesion area of those speckles, and that they also last much longer while the other ones are getting disassembled.
How did you quantify that? How did you measure?
Just interactively. You analyze interactively. You analyze cell by cell picture. Manually, you analyze that. So this is not by a threshold. But you could also do this with a threshold analyzing cytoskeletalizing.
Right, but you distinguish those that are formed by philopodia versus, so it's not enough threshold, right?
Exactly. So it's really that you at first find the philopodia, then in the next step the focal adhesions and so on. So the reason why those adhesions in the philopodia dependent manner might last longer, you can see here on the next slide. Because what you see here is a phylopodium and labeled for the cross-linker you have in those philopodia, a very dense cross-linking of the 20 to 30 actin filaments. But as soon as the cross-linker of the philopodia becomes overgrown by the leading edge, this cross-linker fascin becomes replaced by another cross-linker, alpha-actinin. And this alpha-actinin actually is a cross-linker that is larger and increases the distance between the actin filaments within the bundle. Here you see the same basically in a movie. You have the philopodia, you have here the signal for the alpha-actinin, and only where the lamellipodia is localized you have the alpha-actinin signal, while the extension is not labeled by alpha-actinin. So, and as soon as alpha-actinin becomes incorporated, there is the possibility for other proteins to incorporate into this preformed actin bundle. And here shown in phase contrast, the phylopodium, over time, the phylopodium becomes overgrown by the lamellipodium. Here again, the alpha-actinin labeling the lamellipodial part of this actin bundle, and then again clearly behind the leading edge, then at some point myosin light chain which binds to myosin then also becomes incorporated into those actin filaments, indicating that those actin bundles then become contractile, can generate forces. And this brings me to a traction-dependent adhesion maturation model published here by Schwarz and Gardell. And what you have here in principle is a very early adhesion complex, and it now does not really matter if this adhesion complex is originally located in the phylopodium or in the lamellipodium. the most important thing is that as soon as the young adhesion complex connecting the extracellular matrix to the actin is formed, you get a few adhesions or interactions between the actin and the cytoskeleton. But these very few interactions are not really inducing a lot of force to the adhesion site. You see this here. Now also the tractions that are transmitted by these very young adhesions are very low. And now at some point those adhesions can mature if increased forces are getting applied to those adhesion sites. You see it here schematically. It induces a stabilization, tractions increases simply by binding to actin bundles or to the actin cytoskeleton in general. And as soon as myosin motors are getting incorporated into those actin bundles, then the forces that are getting transferred to the actin are sufficient to induce maturation. And this maturation then induces an opening of various protein conformations. The integrin itself then binds stronger to the extracellular matrix and then inducing the whole formation of a matured focal adhesion, which can then supply to the extracellular matrix very high tractions and at the same time also reduces then again the actin retrograde flow, because here the actin now is getting pulled by the contracting myosin within the actin filaments, and the focal adhesion reduces the traction simply because it strongly binds to the actin filaments. And this model could be actually proven by a couple of experiments. Here, for example, the retrograde flow analyzed by FRAP experiments, so fluorescent recovery after photobleaching experiments, where you bleached into stably adhered actin bundles, so actin bundles bound to a focal adhesion, a line, and follow this line over time, while you have here on the right a filopodium without a stable focal adhesion. And if you analyze this, then over time you can well see that actually really the retrograde flow simply by binding of the focal adhesion to the actin cytoskeleton is clearly reduced within the actin bundle to approximately 1 micrometer a minute, while the retrograde flow within actin bundles without this strong interaction is 2 to 3 times higher. In addition, if this is actually really true, the model, then you should also see a change in forces that are getting applied to the surface depending on whether you have mature or immature focal adhesions. And as you have seen before, you have elastic substrates you can cells grow on. Those elastic substrates also can be labeled with some marker particles, for example, and upon contraction of the cell, the marker particles become deformed. The marker particles, the displacements can be detected and then back calculated into forces. You can see here what's actually happening. If a keratinocyte is migrating over such a surface, the marker particles become displaced on the surface, and this can then be used to calculate for every focal adhesion the forces that are getting applied to the surface. And this is shown here. And what you see is that the forces actually are relatively small in the 0.5 to 1 nanonewton range for very young focal adhesions, and over time then those forces increase the more mature those focal adhesions become. And most or highest forces are actually visible at both rare ends, where there the focal adhesions are most matured. And this is what brings me to the last point, the different aspects that are going on in the lamella. Because what you have heard until now is basically that you have the actin polymerization RQ3 dependent inducing the lamellipodial extension, but within the cell you have those focal adhesions and some actin bundles in between the focal adhesions. Within those actin bundles there's then actin, the myosin contracting the whole system. However, this cell is very well organized in a way, but this cell will never migrate. So what's now causing the migration while having basically the same system in a keratinocyte? And their first experiments, or not first experiments, fascinating experiments I would say, were performed in Sasha's group, where the cells were adhering to a predefined adhesion spot, and then they were analyzed over time in respect of the adhesion sites and also the actin cytoskeleton. And what was found is that there are two specific types of actin filaments that very much differ to what you see to a classical sessile cell. Mainly you have cells or actin filaments that are bound, connected to focal adhesions all around those roundish cells. And those actin filaments, the dorsal stress fibers, direct more or less to the center of the cell. But on the other side of those stress fibers, there's no second additional focal adhesion in contrast to those systems. Instead, you have transversal arcs, and these transversal arcs interact with the actin dorsal stress fibers. And then, in addition, he analyzed the myosin within this system, and
maybe you will leave me.
Okay, so then I'm not showing.
No.
But yeah, so I'm not talking too much about it. But the important thing is this system differs very much to a sessile cell simply because the actin stress fibers are not connected on both ends via focal adhesions. And this is what you actually also have in a migrating keratinocyte. Here you have also those dorsal stress fibers connected at the lamellipodia to the focal adhesions, and then inside the cell body, the lamella, those systems then connect to transversal arcs. And these transversal arcs, you will see this in the next slide, are really different in respect to a classical sessile cell in a way that you have again the focal adhesion here at this point for the dorsal stress fibers. You don't have very much of myosin in those dorsal stress fibers, and then the connection to the arcs spanning the cell from one end to the other side. And in contrast, you have the transversal arcs, and these transversal arcs now are very much filled with myosin. And the more these transversal arcs during migration and also during caused by retrograde flow becomes closer to the rare end, the more myosin actually becomes incorporated. So the very early transversal arcs during migration have still, let's say rarely, myosin. Later on, they are really packed with myosin, and that induces strongest forces at both rare ends, pushing the whole cell body in direction of migration. And this now is shown here in these two movies once again. Here on the left, you see the transversal arcs continuously formed during migration, and the more they reach the end, the more myosin becomes incorporated, which is now not visible in this movie. But you also see.
A question.
Now you don't see anything anymore. Yeah?
So is there additional actin being added to these transversal arcs, or is there something wrong?
So the transversal arcs are mainly formed by the retrograde flow of all the actin that is getting polymerized in an ARP2/3 dependent process in the lamellipodia. And this, by retrograde flow, becomes then incorporated into the transversal arcs that are then getting transferred back.
But they're shortening as you get toward the rear, right?
And they are getting thicker.
And getting, okay.
Yeah. So, and the same, yeah.
But these dorsal arcs contain myosin, right?
The dorsal arcs, especially at the end, yeah.
And then they are putting the cell together, but the cell is preserving its elongate. Or
yeah, you will see this here on the next movie. So this is why. So you mean why is it?
The cell doesn't get narrower.
Yeah. So the reason for this is that this actually takes place for the transversal arcs at the very end of the cell, at the rear end. Instead, the transversal arcs that are still basically at the lamellipodial beginning, they still rarely contain myosin. Therefore, the contraction there is still relatively low. And you see this also here. If you, so my computer now is not working anymore. So anyhow, I'm almost to the end. So what you, so it's crashed. What you should have seen now is that exactly in first place the focal adhesions are getting formed. They're getting stabilized over time. But then that those focal adhesions at the rear end, also the transversal arcs at the rear end, are then getting really pulled inside. So here those focal adhesions really change its position, become located or localized into to the center of the cell, while those focal adhesions grow, and as soon as the cell then overgrows those focal adhesions and the focal adhesions reaches the end, then again those focal adhesions then become pulled to the center. So this is most likely caused by two effects. And I don't know, is it maybe? No, it's not working anymore. By two effects. The first thing is the forces increase due to increased myosin incorporation. But then in addition, the major problem remains, because in principle what the model says, you have largest forces on those focal adhesions at the rear end, and that would mean that they are fully matured, very stable. And this is actually not the case, because those focal adhesions really must disassemble within very short periods of time. And the reason how this might happen is that a couple of proteins become transported to those rear end focal adhesions or become reactivated. So one protein that becomes transported is calpain. And calpain is a protease that was shown to digest specific proteins within those focal adhesions, like paxillin, for example. And in addition, it was shown that proteins become reactivated that induce the protein exchange protein dynamics. So meaning that proteins become phosphorylated in an enhanced manner, and that this induces that in those complexes again a very high exchange dynamics, possibly, and this is again something that would be very interesting to me, then possibly connected with the high forces induces in this case then the disassembly. So and that, yeah, unfortunately brings me to an end, and I'd like to thank.
So again, for the dorsal arcs, does it mean they mature at the end of the lamellopodia, so maybe at the beginning of the lamellum? They move backwards, they're more or less inactive. The more they get to the back, the more active, the more contractive they get. And when they finally at the back of the cell, they contract the cell and push the cell somehow forward.
The transversal arcs. You mean
the transversal?
Yeah.
The
so
transversal.
Exactly. So the transversal arcs are exactly as you mentioned, formed, and then at the end contain more and more myosin.
And that's pushing the cell forward somehow.
Yes. So maybe just one comment once again to Sasha. As you hopefully take with you back home, it is really most important for a focal adhesion formation that it is connected for maturation to actin bundles. And this binding then induces, due to myosin incorporation, forces to those focal adhesions and then causes maturation. And whether those actin bundles are preformed in filopodia or formed later on within the lamellipodia, making a focal complex to a fully mature focal adhesion, I would say is not really the main point.
Actually, focal adhesions nucleate growth of actin bundles by themselves.
Yes, by themselves.
So,
so just looking at the direction of the forces, make me wonder how the nuclei is propelled if it's all sort of in front. If I imagine there's a nuclei at the back of the cell, what makes it move forward if all these arcs are in front?
So if you so all arcs in front are in front in this movie. So in very most cases, here you see the nucleus. You also have very strong arcs here directly
So
behind.
is it enveloped by these arcs?
Yes.
The forces transmitted to the substrate by the cell through the focal adhesions. How does it cause the cell to move forward? Because I thought if the forces are large, that would mean that the cell is more attached to the substrate. So I don't understand how that would make the cell to move forward.
So what you have actually are two different types of forces generated during migration of an epithelial cell. So the first one is really the polymerization of actin pushing the membrane. So this force is in a very low regime, but absolutely sufficient to push the membrane. So then in between this force caused by the membrane polymerization, you have the focal adhesions, more or less separating the pushing forces of the actin and the myosin contractile forces to the rear end starting from the focal adhesion. So, and those forces then, due to this very specific organization of the actin cytoskeleton during migration of epithelial cells, induce this pushing system. If you would have the same forces for a fibroblast where those stress fibers are connected to on both ends to focal adhesions, then this would not induce the migration of the cell.
Is there any understanding about what determines the direction of these filopodia, and do they always stick out in the normal direction to the membrane? If they don't, then those who stick out in non-perpendicular direction, do they have any contribution towards the movement or cell polymerization?
So the first thing what you need to have is the polarization of the cell. Of course, you need to have the lamellipodia, and this is, as soon as it is established, relatively stable due to the localization of membrane-bound GTPase proteins at the lamellipodia. So then there, the filopodia are also getting induced information and are getting formed. If you would have here those cell system, you also get lamellipodia in basically for the whole half circle. But interestingly, the direction of the cell, as long as also the central filopodia are able to bind to the surface, does not largely change. If
always perpendicular to the surface of the cell.
Yes.
All right.
So some of them are not binding properly to the surface, then those are getting pulled back into the lamellipodium, but they are always pushing out perpendicular to the leading edge.
So I guess we stop here and have a coffee break and then come back in 15 minutes.